Hello, for today's video we are looking at microscopes, and we are also going to look at the difference between resolution and magnification, and we are going to also do a couple of calculations to look at magnification and to work out size of objects that you might deal with when working with microscopes. Now, in terms of the actual types of microscopes, we have three key ones that we usually work with. The first one is the light microscope, and this is the one that you have probably used in the lab. You will have seen maybe onion cells, maybe pre-prepared slides of red blood cells, and you may have been able to make out the nucleus in these cells and make out the cell wall and a few details of the cells. Now, the key thing about light microscopes is they can magnify objects to make them look about 1,500 times bigger than they are in real life. So we say the magnification is about 1,500. Microscopes, light microscopes, are fairly easy to use. They can just be brought out, and the substance you're looking at or the object you're looking at can be easily prepared and looked at. Light microscopes use light rays to see these objects, which might seem quite obvious, but they do use light rays. The only problem with using light rays to look at objects is that we can't see objects that are very tiny and that are present in cells. The reason for this is, while we think of the wavelength of light as being quite small, in actual fact, when we're dealing with these very tiny objects, it actually works out being a bit too large for us to be able to see these objects. So if we've got little structures that we're looking at inside the cells, and we've got our light rays with a very large wavelength, we're not going to be able to detect some of these smaller objects. Now, what we can do is, rather than using light rays, we can use a beam of electrons. So we can use an electron beam, and an electron beam has the advantage that it also travels in waves, but the wavelength is much smaller. So if we've got our tiny objects, the electron beam with its much tinier wavelength can actually interact much more easily with these objects, and we can then do a little bit of processing and create an image. So we have electron microscopes that use beams of electrons with a smaller wavelength than the wavelength of light. And the two types of microscope we tend to look at are the scanning electron microscope and the transmission electron microscope. And there are a couple of details you need to know about both of those. With our scanning electron microscope, or our SEM, we have electrons that are reflected off the surface of an object. That gives us a 3D image, and we can work with magnifications of around about 100,000 times. So real life objects look about 100,000 times bigger than they actually are in real life. The good advantage for this is, obviously, we get a 3D image. Sometimes we add color to make images look a bit clearer, but that's our SEM. The transmission electron microscope, we have electrons passing through the object. That means when we're looking at samples, they have to be very, very thin. It creates 2D images, but the magnification is much higher. It goes up to about 500,000 times magnification. The one key disadvantage of using a transmission electron microscope is there is a lot of preparation of the sample. You actually have to have training in order to be able to prepare your samples, and it takes quite a while, and you have to be very careful, and sometimes you do a lot of preparation of the object and it doesn't work out that you see very much at all. So you have to maybe have another go. But really, basically, it takes a bit more effort to be able to create images with our transmission electron microscope. We have, however, managed to get a lot of detail about the structure of cells using these two kinds of microscope, and we'll look at that in more detail in a future video. What I want to do next is to make sure you understand the kinds of units that we use when we're working with tiny objects such as those that are present in cells. We tend to deal with micrometers and nanometers, and we tend to use millimeters less, but the relationship between micrometer, nanometer and millimeter is as follows. The way we write it is, for our millimeter, it's two small m's. Our micrometer has a funny shaped U and an M, so that's a micrometer, and nanometer is an N and an M, nice and simple. If you want to convert between the three, which you may well need to do, to go from millimeter to micrometer, simply a case of multiplying by 1,000. If you want to go down further to a nanometer, we multiply by 1,000 again, and if we're going in the opposite direction, it's dividing by 1,000. So if I gave you an example, I don't know, if I said to you an object is 2.5 millimeters, what is that in micrometers? Simple case, multiply by 1,000 and you get 2,500 micrometers. Okay, and you can use this idea here to convert between the units, which, as I say, you may well need to do. The next thing to look at is this idea of magnification versus resolution. Magnification is usually quite straightforward to understand. It's just how many times bigger an object is, an image is, than the actual object. So I don't know, if I've got an object here in real life, I manage to get an image that is that big. You can see that this is roughly 4 times bigger, so we would say the magnification here is times 4. It's been magnified to a size 4 times bigger than the actual object. That's quite straightforward to understand. Resolution, however, is a little bit more tricky. Resolution basically gives you the detail in images. It gives you sharper and clearer images, and what it means is the smallest distance which 2 objects can be seen has to be separate objects, or in other words, the minimum distance that 2 objects can be in order to appear as separate. So really the everyday English way of explaining it is you see more detail, you see sharper and clearer images, but that's the slightly more technical way of explaining it. Let me show you an example of how that might work. So here I've got an actual object. If I was to magnify it, can't see it very clearly there, but if I was to magnify it, this is a high resolution image of what we were looking at. You can see 2 distinct different shapes. These could be pollen grains or something like that, but you can definitely see there's 2 different ones there, and you can see a nice clear outline of those 2. If I had the same magnification but with poorer resolution, here's the same magnification, magnified by the same amount, but the resolution is very low. So here you can just about make out the outline of the 2. You can't really tell if they're a separate object or the same object, and that's because the resolution is quite low. So these are 2 images, one with low resolution, one with high resolution, and that gives you an idea of what we mean by magnification versus resolution. So for our electron microscope, the resolution is about 20 nanometers, so very, very tiny. Objects can be very, very close and we can still see them as separate objects, and not only that, we can magnify by a huge amount as well. Whereas something like a light microscope, the resolution is only about 20 micrometers, not 20 nanometers, and the magnification is about 1,500. So as a result of that, electron microscopes have enabled us to see very sharp and clear images of very, very tiny things. The last thing I want to do for this video is to work out 2 things. The first thing is magnification, and the second is the size of an object. In order to work out magnification, we need to know 2 bits of data. We need to know the size of the image that we have, and we need to know the size of the object that we have, and the formula is basically size of image divided by the size of the object. So let me explain what I mean, or in fact give you an example. Here we have a mitochondrion, and we want to look at the magnification based on the distance given by this red line. So to work out the magnification, we need the size of the image, and we need to know the size of the object. Now the size of the object is actually given here. It's 10 micrometers, but we need to know the size of the image. Now there's one of 2 ways we can get this information. Either it's going to be given to you in the exam, or we would need to get a ruler and measure it ourselves. So if I get a ruler here and look at that distance, it's 3.5 centimeters. Let's call that 35 millimeters. So I've measured that as 35 millimeters, and in terms of the size of the object I have 10 micrometers. So the substituting these numbers into the formula that we have here, we would have 35 divided by 10 micrometers. But you will notice straight away that these two are in different units, and we can't work this out if they're in different units, so we would have to make sure the units were the same. And in terms of doing that, 35 millimeters is probably quite straightforward to work out in micrometers if we use our method that we had before. So we've got 35 millimeters, and we want to go to micrometers, and all we do is times by 1,000. So we have 3,500. No, we don't. We have 35,000 divided by 10. And if we work that out, the answer is 3,500. So the magnification for this object is 3,500. The image is 3,500 times bigger than the actual object. And we have no units, as we know, and that there is the answer. Now what if we want to work out the size of the object? We have the magnification and we have the size of an image. Well, let's have a look at an example for that one. So here is a cell. It could be a bacterial cell or something like that. We have a bit of data in terms of the magnification, and we also have the size of the image, which is 20 millimeters. There's two ways we could do this. In terms of the size of the image, again, we could either get our ruler and measure it, sticking out from the top of the screen, and let's get rid of that. We can use our ruler to measure it, or we can actually, or it will be given to you in the question. So for this particular example, it's been given. So what's the size of the real object in real life? The size of the image is 20 millimeters. And it's been magnified by 100,000 times. So we divide that by 100,000, and the answer we get is 0.0002 millimeters. Now, what if the question said give the answer in micrometers? Well, we've got 0.002 millimeters. Well, we can use our diagram that we had before. We've got 0.0002 millimeters, and we want to know what the answer is in micrometers. So we basically multiply that by 1,000. And if you're quite good at maths, you can do this without a calculator, and that will give you an answer of 0.2. So going back, the object in real life is 0.2 micrometers. Okay, so there's two examples of how you would work out either magnification or the size of an object, and that's basically all I wanted to go through for this video. So thank you for watching. I'll see you again soon.